Hello, everybody. In this video lecture, we will cover chapter 14, sensory physiology. So let's go ahead and begin. So sensory neurons, so we call them receptors. Each sensory neuron responds primary to one particular type of stimuli that is called its normal stimulus. For example, photoreceptors are activated by light. However, they can be activated by pressure, but it would not be considered a normal stimulus. The dendrites of the sensory neurons transduce or convert the stimulus into changes in membrane potential. So when we apply stimulus, this increases permeability of cell membrane to sodium ions. So sodium, we know we have high concentration of sodium outside cells. So sodium start moving inside. So positive charges moving in, it causes depolarization of dendrites. And this is called receptor potential. And if it reaches threshold, then at the axon hillock, action potential can be generated. And action potential is a long distant communication signal. Continuous stimulation of the sensory neurons result in decreased response. That is called adaptation, right? So for example, if we say, let's say, if you constantly smell something, right? If you enter a room with a very bad smell, first it's a very strong odor, but then you stop feeling that smell. This is because olfactory receptors are very quick to adapt. Or another example would be if you put glasses on the top of your head and then you start looking for your glasses. Well, actually glasses, they apply some pressure and you should feel this touch and pressure, but because receptors adapt, you stop feeling this pressure. Some receptors, they either never adapt, like pain receptors, because we don't want to be used to pain, or proprioceptors, that's receptors about the movement of your body parts and location of your body parts in the space. Now, sensory neurons can be classified based on the different themes. For example, this one is based on Charles Sherrington, who got a Nobel Prize for his classification, and his classification was really very straightforward. He divided sensory neurons into somatic sensory neurons, and that means sensory information that is carried by those neurons reaches your consciousness. In visceral sensory neurons, information does not reach consciousness or is only poorly defined and localized. And visceral sensory neurons, they bring information about sensation to your brain from your internal organs, from your viscera. Also, we can classify sensory neuron based on their structural classification. They can be either simple receptors or complex receptors, and a receptor for special senses. So simple receptors, they are just free nerve endings. So those are just dendrites. Complex neural receptors, they are also dendrites, but they have connective tissue capsule surrounding them. And special senses, those are special cells, such as rods, cones, hair cells for hearing, equilibrium. So they are found in a special senses organs. Also, we can classify receptors based on a stimulus type, like chemoreceptors. That means their stimulus is some chemical, change in the chemical composition. Mechanoreceptors detect physical changes in the tissue, touch, changes in the muscle length, joint angle. Thermoreceptors detect temperature in changes in temperature, right? And proprioceptors. Proprioceptors are also another category. I don't know if it's included here or not. In some books, you would have the special function proprioceptors, but over here, you know, they just in the mechanoreceptors category. Photoreceptors. Photoreceptors detect light in eyes, rods, and cones, right? So that's classification by stimulus type. So over here you can see on the top, two types of receptors. They are actually dendrites, right? So this is a simple and this is complex. Those are free endings. This is encapsulated dendrites, and this one is rod that is a specialized receptor cells for vision. So here we have simple, complex, and special senses, right? So that's our simple, those are complex, and those are special senses. Now, sensory pathway general pattern. Sensory nerve fiber that we call first order neuron synapses into myelinated interneurons that we call second order neuron in the central nervous system. Right, so we have first order neuron and second order neuron, and then we have third order neuron. So we need kind of like in general pattern, we need 3 neurons for sensory information to go from periphery to your brain. Second order neuron decassate, that means it's moving from left to right, from right to left. It travels into bundles upward through central nervous system, synapse in the thalamus into myelinated third order interneuron, and the third order neuron projects to many areas of the brain, including the cerebral cortex. Right, so over here, you can see how this person feeling pain. So we have nociceptor, that is a pain receptor, and we have first order sensory neuron, right? That synapses in the spinal cord with the second order sensory neuron. Second order sensory neuron decassate. So you see it moves on the opposite side of the spinal cord, and it goes up through the spinal cord. It runs in a bundle of axons that are called ascending tracts. And then it synapses in the thalamus with the third order neuron, and a third order neuron sends the sensory information to the different part of your cerebral cortex, right? For example, over here. Integration by central nervous system. So sensory information travel from spinal cord to brain by ascending pathways or directly to brain stem via cranial nerve. Visceral reflexes integrated in brain stem or spinal cord usually do not reach conscious perception. So if we have reflexes that do not reach our cerebral cortex, we usually do not understand that sensation. Right, so here you can see how a different sensory neurons bring information to the cerebral cortex, right, or cerebellum, right? So number 1 is olfactory pathway from the nose. This one reaches your cortex before it goes to the thalamus. The rest of the sensory information goes to the thalamus first and then to the cerebral cortex. Right, so most sensory pathway project to the thalamus, right? And equilibrium pathway projects primary to the cerebellum. So cerebral cortex is divided into several functional areas, right? We know that in anatomy, you learn that it's divided into lobes like frontal, parietal, occipital, temporal. There is also insula. Another lobe, this is cerebellum. But if you look at functional parts of the cortex, so over here we have primary sensory area that is in the parietal lobe. It's called post-central gyrus because this is central sulcus, this line. So it's post-central gyrus is a primary somatosensory area, and pre-central gyrus over here, it's a primary motor area. Then we have premotor cortex, right? We have prefrontal cortex over here, where your personality is, right? Your higher intellectual function. We also have in the occipital lobe primary visual area, vision association area, in the temporal lobe primary auditory area, and auditory association area. So a different type of sensory neurons brings their specific signal to appropriate part of the cerebral cortex. Now, definition of pain. Why are we talking about pain? Because pain, it's also sensory information. So it's unpleasant sensation associated with suffering. Pain is the only sensation that has a build-up emotion. We all agree how we feel about pain. So nociceptors are special receptors for pain. They respond to strong, noxious stimulus that can damage tissue. And for pain receptors, we have free nerve endings, so free dendrite, and primary sensory fibers. So this is the pathway for pain. Noxious stimulus, right? Injured tissue cells release chemicals, right? So when we apply stimulus, we damage those cells. Those cells start release inflammatory chemicals. Kinins, prostaglandins, those are example of those inflammatory chemicals. And they stimulate nociceptors. They send signal about pain into thalamus and limbic system. Limbic system, this is where you have your emotions, right? That's how you feel bad, right? You feel this suffering. And thalamus gonna send this information into right part of your brain, right? And then from thalamus and limbic system to cerebrum to allow us sensation of pain. Now, referred pain is when we have visceral internal pain that is generally projected back to the surface of the body. Like your heart hurts, but you feel like you have pain in your shoulder, in your left shoulder or in your left arm. And the basis for it is that visceral nociceptors, receptors for pain, they converge with somatic sensory receptors. And that's why your brain confuses. It cannot really localize and define precisely where this pain is coming from. So example would be ischemia, that's the lack of oxygen flow in your blood. And chronic pain is pathological neuropathic pain. So that's not referred pain, it's just a chronic pain. So here you can see how, let's say, liver and gallbladder, right, can be projected to the shoulder, how heart pain or ischemia can be projected to the shoulder again and the arm, right? How pain in the uterus or pain in the colon projected to the skin. So the patient feel like pain comes from the skin, from muscles, but not from internal organ. That's what referred pain is. And this has happened because, look, this is a pain receptor or nociceptor from internal organ. So this is a visceral sensory receptor, and this comes from skin, right? And this is somatic sensory receptor. And you see how they synapse to the same secondary order neuron, right? So pretty much your brain just like, okay, it's coming from this neuron, so your brain confused and think it's coming from skin. Now, a pain modulator are analgesic drugs like aspirin, Tylenol, or Advil. They inhibit inflammatory chemicals, and such as prostaglandins, right? And that decrease inflammation and slow transmission of pain to site of injury. Opioids, opioids, they decrease the processing of sensory information about pain in the thalamus and the limbic system, includes endorphins, morphine, Vicodin, lidocaine, xylocaine. They are other local anesthetics and they block the conduction of action potential in nerve fibers. So we do have some drugs that can modulate your pain, right? So either they decrease inflammation or they interfere with the processing of sensory information to your brain. Okay, and we will only cover vision briefly and hearing in this chapter after we cover this general information about sensory receptors. So structure of the eyeball. Walls of eyeball contain 3 layers: fibrous, vascular, and sensory. And there is internal cavity that is filled with fluids called humors. The lens separate internal cavity into anterior and posterior segments, right? So here you can see the cross section of the eyeball, and of course it's diagrammatic view. And here's the lenses, and lenses separate this internal cavity into anterior segment and posterior segment. And inside those segments, we have humor. Humor means fluid. And in anterior segment is called aqueous humor, and in posterior is called vitreous humor. Also, you can see 1, 2, 3 layers of the eyeball over here that we will talk about in more detail right now. And what exiting the eyeball over here, that's an optic nerve, right? So that's an optic nerve. So fibrous layer. If you look at this diagram here, it's shown in blue and green color. It's an outermost layer, dense avascular connective tissue that has 2 regions, sclera and cornea. And sclera is that white part, right on the posterior region. It's opaque posterior region. It protects and shapes eyeball. It anchors also extrinsic eye muscles. And cornea, shown here in green color, so it's also part of the fibrous layer. It's transparent, anterior, 1/6 of fibrous layer. It bends light as it enters the eye. It has sodium pumps that keep it clear, right? And it has numerous pain receptors contributing to blinking and tearing reflexes. So fibrous layer, cornea, and sclera. Middle of vascular layer, uvea, has 3 regions. So shown on this diagram in 3 different colors: red, yellow, and purple. So it's a choroid, ciliary body, and iris. So choroid region, posterior portion of the uvea, supplies blood to all layers of the eyeball. No wonder we call it vascular layer. And it has brown pigment that absorbs light to prevent visual confusion. Then ciliary body, shown here in yellow color, it's a ring, right? Because that's our, this is mid-sagittal section, right? So you kind of see like pieces of it, but it's actually a ring. Ciliary body is a ring of tissue surrounding lens. So here's the lens and ring surrounding the lens. It's a smooth muscle bundles, ciliary muscles that control lens shape. Capillaries of ciliary processes, so they have like this little like finger-like structure called ciliary processes. And inside those processes, we have capillaries and they secrete fluid. Ciliary zonules are ligaments that hold lens in the position. Let's see if we have it or not. Let me see if I go back over here so you guys can maybe see it. Yeah, let's look over here. So you see this is ciliary body. Then those kind of like finger-like structure, ciliary processes, but those lines, like blue lines, those are suspensory ligament or ciliary zonules, and they hold lens in place. And they also, when they stretch or when they relax, they regulate the curvature of the lens, right? Lens can be more flat or less flat, right? So that's those ciliary zonules. right? So that's ciliary body. And iris. Iris is the color part of the eye. It's also vascular layer. Now, pupil is just an hole, just openings that allow light to enter the eye. Now, when we have a close vision and bright light, then pupil constrict, right? Well, it's a bright light, right? We're looking close. We don't need much light to enter the eye, so pupil constrict. Now, when we have distant vision, when we look far and a dim light, then the pupils dilate. Also, changes in emotional states dilate the pupil and also make it kind of appealing. So when we have like a dinner and we have candles, in this kind of dark light, our pupils dilate and it's look appealing, it's look pretty, right? Now let's look how this has happened. So, so here's your pupil. This black hole, right? That's an opening into eyeball that allow light to enter your eyeball. And we have two sets of muscles. We have a sphincter pupillae muscle and we have dilator pupillae muscle, right? So one is sphincter. Sphincter is always circular muscle and dilator. So they both contract, right? But when sphincter contract, then the pupil constrict. When dilator contract, it's get, you know, shorter, right? Then the pupil dilate, right? And sympathetic nervous system, when we under stress, then our pupils dilate. When we relaxing, then parasympathetic nervous system controls the size of the pupil and the pupil constrict, right? So you can see over here on the iris, right, that has this nice bluish green color. And actually the iris is made of two sets of smooth muscles. Now, then we go to the sensory layer, right? So we already covered, we have fibrous layer, right? That's a fibrous outermost. Then we have vascular layer in the middle. And then the last layer of the eyeball, we have sensory layer or retina. Retina is delicate two-layered membrane. It has pigmented layer and its outer layer, right? So here's the pigmented, just a little line over here. And then it has, well, this pigmented layer, it absorbs light and prevents scattering, and it also stores vitamin A. And then it has neural layer, right? So sensory layer or retina has pigmented outer layer and neural layer. Now, neural layer has photoreceptors that transduce light energy. So they transform light energy into action potential energy. So cells that transmit and process signals are, so it means the neural layer is made of bipolar cells, ganglion cells, amacrine cells, and horizontal cells. So let's look at them over here. So you can see first pigmented layer of the retina, and this one like epithelial cells. And their job is to absorb light and doesn't allow it to reflect back. And then we have neural layers. So we have photoreceptors, rods, monomers, and cones. Then these cells over here, bipolar cells, and then ganglion cells. And ganglion cells, they have long axons, and their axons make this optic nerve. Also, we have amacrine cells and horizontal cells that just allow communication between these three layers of cells. So ganglion cells, axons, run along the inner surface of the retina and they leave the eye as the optic nerve. Now, photoreceptors, rods, more numerous at a peripheral region of retina, away from central part that is called macula lutea. They operate in dim light, provide indistinct, fuzzy, non-color peripheral vision. And cones found in the macula lutea, and macula lutea is a special part of the retina where you have your sharpest vision. They concentrated in the fovea centralis. Fovea centralis right in the side of macula lutea. Macula lutea means yellow spot. Fovea centralis is right inside this macula lutea. Cones operate in bright light and they provide high acuity color vision. And we have 3 types of cones, red, green, and blue. So they respond to different wavelengths of the visual light. So red, they activated by red light, green by green light, blue by blue light, and then, different degree of activation and different combination of activation give us a whole spectrum of colors. Okay, now pathway of light entering the eye. So we go to the cornea, then aqueous humor, then lens, then vitreous humor, the neural layer of retina, photoreceptors. And then from photoreceptors, from photoreceptors, they create this, receptor potential, and then ganglionic cells, they create action potential that leaving, your eyeball through optic nerve. So light is refracted. Where it's refracted? At the cornea, entering and leaving the lens. And that's why when we change the curvature of the lens, it allows us to focus an image. Now, refraction is bending of a light ray due to change in speed when light passes from one transparent medium to another. Light passing through a convex lens, the ones that you have in your eyes, is bent so the rays converge at a focal point. The image formed at a focal point is upside down and reversed right to left. Right, so you can see over here, if you see image over here, right, so this image, because light is reflected, right, and it's upside down and reversed. Focusing for distant vision. Light rays from distant objects are nearly parallel to the eye, and that's why they need very little refraction beyond what occurs at rest eye. So pretty much when you're looking at distant object, you don't need to focus much. You can just, your eye just can be in this resting stage, right? Because rays are almost parallel. Now, far point of vision is the distance beyond which no change in lens shape is needed for focusing. So 20 feet is for normal or emmetropic eye. So all the objects that are beyond 20 feet, right, you don't need to focus. So you don't need to change the curve of your lens, right? Your eye is relaxed. Does it make sense? So you do not change shape in lens if you're looking at something that is beyond 20 feet. This eye, of course, if you have normal emmetropic eyes. And then ciliary muscles, remember those ciliary muscles that are circular muscles that are relaxed. And when they are relaxed, length is stretched. So lengths are flat by the tension of the ciliary zonules. Let's see if I have it, right? So let's look over here. So we're looking at a distant object over here, right? So, and from the distant object, those rays are almost parallel, right? So you don't need to reflect them. So you don't need this angle to be very sharp, right? You don't need lots of refraction to focus it on the retina. Then the ciliary muscles are relaxed, right? So ciliary muscles, look, when let's say ciliary muscles relax like this, what happens when they contract, right? Like this. So they, because they're circular muscles. So when they relax, they like that. When they contract, the diameter decreases, right? So here they are relaxed. And when they relax, those zonules, they are stretched and they keep lenses flat, right? But ciliary muscles relax, zonules are tight and lens are flat. Now, when you need to focus for close vision, we call it accommodation, right? So again, do you need to focus for far vision? If it's beyond 20 feet, no, not really, right? But when it's get closer than 20 feet, then you need to change the shape of the lens. And this is called accommodation. Okay, so what is the nearest point of vision? It's determined by the maximum bulge that lens can achieve. So your lens can only be, as concave as they, supposed to be. That means you cannot see anything that closer. If this bulge is not enough, you will not be able to clearly see objects that are closer than that. Now, presbyopia. It's a loss of accommodation over age 50. It's a normal process. That means when we get older, we cannot see close object very well. When we get older, we see far object more clearly than close object. That's why when you're reading something, when you're older, you need to keep it a little bit further away from your eyes. Now, that's a presbyopia. It's a normal loss of accommodation when we get older. Our whole life we had a normal eyes, right? And now we start just have this, hard time looking at close objects. That's why we're buying these reading glasses, right? So that's accommodation. Accommodation is a changing the lens shape by ciliary muscle to increase refractory power. Constriction also required for close vision. It's accommodation pupillary reflex that constricts the pupil to prevent the most divergent light waves from entering the eye. So for the close vision, remember your pupil is constricted. For far vision, your pupils are dilated. And convergence is another requirement for focusing for close vision, and it's a medial rotation of eyeballs towards the object being viewed. So you kind of like, oops, your eyes converge to the midpoint. Right, so here when we are looking at the close object, then rays are they diverge from each other. So they're not parallel, they diverge. So we need really lens to bulge to be able to focus this object on a retina. Now what happens then? Then these ciliary muscles, they contract. So ciliary muscles relax like this, so now they contract. And because they contract, these ciliary zonules, look how they have like this line like this. So they relax, and when they relax, they cannot stretch the lenses, and lenses are not flat anymore, so lenses bulge, right? This is a parasympathetic activation because when we're looking at close object, muscles are activated by parasympathetic system. So we're relaxed, we don't need to look for danger that comes from far away, we're just looking at close stuff. Problem with refraction. So myopia is nearsightedness when the focal point is in front of the retina. So actually object like focal point, you're focusing stuff in front of the retina. And this is because most of the time, because eyeball is longer than normal. It also can be because of the problem in the curvature of the cornea, right? Maybe some problem with lens, but most often, right, the myopia, if somebody is born with myopia, right, then the eyeball is longer than normal, an image focusing in front of the retina. And it can be corrected with concave lenses. Hyperopia or farsightedness, when focal point is behind the retina, and it's because the eyeball is too short. And it's corrected with convex lens. Astigmatism caused by unequal curvature in different parts of the cornea or lens, and corrected with cylindrically ground lenses, corneal implants, or laser procedures. Right, so this is normal emmetropic eye. So here we have light rays, right? We have them refract and focusing right on the retina, right? So that's all normal. Now, what we have myopic eye, remember myopic eye, eyeball is too long, and the focal point is in front of the retina, right? So nearsightedness, it's focusing too close, right? It doesn't reach the retina. So then we use concave lens, right? So we decrease this refraction. So you see how we first diverge those are parallel, right? So we diverge those rays, and then we convert them back right on the retina. Now, when it's hyperopic eyes, then the focal point is behind the retina because eyeball is too short, and this can be corrected with the convex lenses that moves focal point forward. Okay, so that's it for vision. Ear, hearing, and balance. There are three parts of ear: external ear or outer ear, middle ear, shown right here, and it's called tympanic cavity, and internal or inner ear. So external ear and middle ear are involved with hearing. So external and middle with hearing. Internal ear function in both hearing and equilibrium. So receptor for hearing and balance. So you have receptor for hearing and you have receptor for balance, right? Or equilibrium. They respond to separate stimuli and they are activated independently. So this is transmission of sound from the external ear, right, to the internal ear. So, so here we have number one, sound waves vibrate tympanic membrane. So here's the tympanic membrane. Sound waves are mechanical waves, so they just vibrate, right? Now, in the middle ear, we have what we call auditory ossicles. Those are little bones that vibrate because of this vibration of tympanic membrane. And what they do, they amplify pressure, right? Now, those pressure waves created by the, so here we have malleus, incus, and stapes, three auditory ossicles, three auditory bones. The smallest one are stapes. So stapes are connected to another membrane. It's called oval window. So those pressure waves that are now amplified, they passing from malleus to incus to stapes, and stapes pushing on this oval window and move these pressure waves inside the inner ear. Now this number three is called scala vestibule. So we have kind of superior, this is called scala vestibule, right? We have inside called cochlear duct, and here shown here beneath, right? The cochlear duct is scala tympani. So scala vestibule, cochlear duct, and scala tympani. Now, these pressure waves start moving through this scala vestibule. Now, if we have frequencies of waves that are below hearing, so we cannot hear them, That means they travel all the way to the end, right, of the cochlea, and it's called helicotrema, right? So this part is called cochlea, right? So in helicotrema, it's the upper part of the cochlea, and if the waves just move just around it, then you cannot hear that sound. But sounds in a hearing range go through the cochlear duct, as shown here in 4B. And what they do, I'll show you in a second. There is a basilar membrane over here, and they vibrate this basilar membrane, and a basilar membrane cause the hair cells, because we do have like some hair cells, the hair to bend, and this bending stuff pretty much, you know, allowed to generation of action potential. Electrical impulses generated when hair cells bend. Right, so so high frequency sounds displace basilar membrane near the base. Medium frequency sounds displace basilar membrane near the middle, and low frequency sounds displace basilar membrane near the apex. And then, so over here, so that's our cochlea, right? So we have our 3 scala, right? Scala vestibuli, cochlear duct, and scala tympani. And over this part over here, this is the basilar membrane. And this whole part here is called spiral organ of Corti. And in spiral organ of Corti, these hair cells, they have hair, right? And when basilar membrane, you know, moving up and down, this membrane over here, right? It's bend the hair cells, and that create the electrical impulse, action potential. So action potential from hair cells move through the nerve, right? And this nerve is, okay, so we have vestibular cochlear nerve. So this is a part of the cochlear nerve, and then it's converged with vestibular nerve, making vestibular cochlear nerve. So it's right here, vestibular cochlear nerve, and it moves to the medulla. Then it moves to midbrain and to the thalamus, and from the thalamus to the primary auditory cortex in the temporal lobe. Let's see if we have, so we do not, okay, so I did not put any really structure of this organ of Corti. I just want to tell you that when you look over here into the inner ear, right? So these structures that look like a snail, that this is a cochlea, right? So if you look at this cochlea, kind of like, because it has those like a, it's a kind of like a spiral, right? And if you stretch it up, so this is how cochlea would look like, right? It would have this scala vestibuli, cochlear duct, and scala tympani, right? And in a cochlear duct, you have a this basilar membrane, right? It's kind of like shown over here, right? And a basilar membrane is a part of the spinal organ of Corti. And spinal organ of Corti is a sensory organ, right? That has receptors for hearing. Those receptors for hearing, they create receptor potential, right? And they send this, well, in this receptor potential, generate action potential that moves toward your cerebral cortex. Now, 2 types of deafness. We have conduction deafness and sensory neural deafness. So conduction deafness, when we have blocked sound conduction to the fluids of the internal ear. So anything that prevent this energy of the sound, those sound waves, to reach your internal ear. So it's just not conducted. So it can result from impacted earwax, perforated eardrum, otosclerosis of those ossicles, so ossicles do not move. Sensorineural deafness is damage to any neural structure at any point from the cochlear cells to auditory cortex, right? So either the damage is in the neural structure or the damage is in external and middle ear, and we just cannot conduct those sound to the cochlea. Tinnitus is imbalance of hearing, and when patient hearing this ringing or clicking sound in the absence of auditory stimuli, and it's due to cochlear nerve degeneration, inflammation of middle or internal ear. It can also be side effect of aspirin. Meniere's syndrome is a disorder that affect cochlea and semicircular canals for equilibrium, and they cause vertigo, nausea, and vomiting. Okay, so so far we were talking about hearing. For hearing, we looked at this yellow part that is cochlea, right? So cochlea is for hearing, but in internal ear, we have two more parts, shown here in green, vestibule, and in blue, semicircular canal, right? So this is our vestibular, so this is a part of the internal ear, right? So vestibular apparatus, however, specifically consists of vestibule and semicircular canals. Vestibular receptors, so this vestibule is for static equilibrium. And static means anything but rotation. So static equilibrium means when you close your eyes, for example, you're sitting in a car, you close your eyes, you're sitting in a car, and cars start moving, but you still have your eyes closed, but you feel this movement. It's called linear acceleration. So you feel this linear movement. So that's static equilibrium, and this is job of vestibule. So when you're in a car with your eyes closed and cars start moving, that's the vestibule that allow you to feel this movement, right? A semicircular canal is for dynamic equilibrium, and this is for rotation. So when somebody just spin you around and you feel, and you know that, you know, somebody is spinning you, that's a receptor inside semicircular canal responsible for that sensation. Right, so so here's our vestibule, right? And those are semicircular canals. So vestibule and semicircular canals, those are bony structure. Then inside we have membranous structure, and inside those membranous structure, we have sensory structure. Now inside the vestibule, we have two special membranous structure. Is called utricle and saccule, right? So again, vestibule is made of bone. So vestibule is bone. Inside this bone, we have two membranous structure that called utricle and saccule. And inside utricle and saccule, we have sensory organ that is called macula, right? So a macula is made of different type of cells, and some of them are those hair cells. So on the top of these hair cells, we have gelatinous matrix, and on the top of it, otoliths. So those are like a little, little stones, right? Little rocks, stones. And when they move, right, when you move, those otoliths move, and they bend these hair cells. And when hair cells are bending, right, we create receptor potential, right? Because again, sodium channels open, oops. And now semicircular canals, again, they are bony structure. Inside we have semicircular ducts, membranous structure, and inside we have sensory organ that is called ampulla. And I'm sorry, yeah, ampulla is a membranous structure, and crista is sensory. So crista in ampulla and macula in utricle and saccule. So just remember, utricle, saccule, ampulla are made of membrane, and macula and crista, those are sensory part, and they have these hair cells. And on so we have hair cells that cover by the gelatinous structure again that called cupula. And when you're rotating, this kind of like cupula rotates and it bends these hair cells. And again, these hair cells create receptor potential and then action potential. And action potential reaches your brain. So you know, you feel your linear or dynamic equilibrium, static or dynamic equilibrium. Makes sense? Okay. So macula. Macula is sensory receptor for static equilibrium. Why? Because macula is located in utricle and saccule in this part that is vestibule, right? So vestibule is for static. You go back or front, back, right, left, up, down, right? So that's all the job of the macula to sense this movement. It also monitors the position of head in a space necessary for control of posture, respond to linear acceleration forces, but not rotation. Now, crista ampullaris or crista is sensory receptor for dynamic equilibrium, one in the ampulla of each semicircular canals, and we have 3 of them. A major stimuli are rotatory movement, right? So again, so those are semicircular canals. Now they have this ampulla, and inside the ampulla we have crista. Right, so here you can see how when this, so that's the whole organ is called crista, that made of hair cells and cupula. And you can see when you're rotating, this cupula bend those hair cells, and this create electrical impulse that moves to your brain. That's the last slide. Okay, so that was chapter 14. Thank you for watching, and I hope it was helpful. Let me just now pause this recording. It's not that easy to do. Okay, try again. Pause recording.